Okay, let's have a class. We will talk about DNA replication today. Before that, we would love to go over something for the review. Okay, I would love to spend a little bit of time, go over aerobic respiration, but briefly. Okay, here's something. If you do not need extra points, which means you're doing very well in the exam 1, exam 2, and you also haven't learned biochemistry yet, you said, I don't need those points. Then what you need to know. Okay, this is the basic you need to know. Glucose going through glycolysis become pyruvate. And the pyruvate going through a transition step become acetyl-CoA. And acetyl-CoA is going through a big cycle called the TCA citric acid cycle. Okay, then we're going to calculate how many ATP from there. Complete aerobic respiration. So you need to know, at the beginning here, we using 2 ATPs. And then, in the branch of the glycolysis, because there is one NAD reduced to NADH2 right here. And then they also generate 2 ATPs going through substrate level phosphorylation. And this indicates is 3 ATPs. Okay, so they add up together is 5 ATPs. Because we have a 2 chain, so they are multiplied by 2. So equals 10 ATPs. At the beginning, we are using 2 ATPs. So minus 2 ATPs, add 10 ATPs is equals 8 ATPs. Going through complete aerobic respiration. And then, going through pyruvate to acetyl-CoA, there is NAD reduced to NADH2. That is 3 ATPs. And in the TCA cycle, you need to know somewhere here, there is happened twice NAD reduced to NADH2. Here, one more is connected to it, NAD to NADH2. Right here happened is FAD reduced to FADH2, and right on the top there's another NAD reduced to NADH2, and do not forget there is a substrate level phosphorylation, which is GDP becomes GTP. Okay, so how many ATP are there? This is 3, 1, 2, and 3. So how many right here? 1, 2, 3. It is 3 multiplied by 3 ATPs from NADH, reduced to NADH2, add 2 ATPs from, I'll just write here, NAD reduced to NADH2, and this is FAD reduced to FADH2. And then we're going to add one more, is substrate level phosphorylation, that is GTP. So you add up them together, how many? This is 12 ATPs. And remember, these two add up together, you need to multiply by 2. Because each pyruvate is going through a cycle right here, and they also going through a cycle right here. Okay, so you add up them together. This is 30 ATPs. Going through 2 cycles of citric acids, and you add 8 ATPs here. So the total is 38 ATPs. And remember, this is for bacteria, we mentioned. Okay? And for eukaryotes, is 36 ATPs. And for microorganisms, we'll talk about is fungi. Yeast and molds, because there is a 2 ATPs is passing through mitochondrial mixture, the barrier, like your paid ticket. Okay, so this is the one, is basic requirements in the class. If you don't know these things, which means you don't have to, because it's not a biochemistry class. But if you need extra points, make sure you're understanding the chemicals of here, structure, chemicals here, structure, and the chemicals in the TCS cycle and the structure. Like I said, one chemical, 0.5 points. Structure, 1.5 points. So there's a lot. Okay, lots of extra points here. So this is something we mentioned about that. Okay, next, we want to also briefly go over what we talked about already. When we go for the genetics part, we are talking about essential dogma. So that means is DNA going through replication, and then DNA will become RNA, and that is called transcription. And then RNA will become protein. This is called translation. And our outline will be follow all these, and today we'll focus about the DNA replication. And if we have time, we'll get to RNA transcription. And basically on Thursday, we'll talk about transcription and translation. And then we talk about this whole thing. Before the central dogma comes out, the people say it's a nucleic acids, go protein. It is not really this procedure. Okay, at the beginning, people talk about nucleic acids, go protein. Then during the history, there's several important experiments lay out the foundation for the structure of the DNA. If you still remember, we talk about 1928, F. Griffith, the Streptococcus pneumoniae in mice study. Okay, this tells us there is some genetic information there. However, we don't know whether it is DNA, RNA, or even protein or other chemicals. Then in 1942, we have Avery, McCarty, and McLeod did a tube study, which is in a bacterial media. We confirm the genetic information is DNA, and it goes through transformation from smooth Streptococcus ammonium transferred to the rough Streptococcus ammonium. Then we talk about 1952, basically, okay? We have like Hershey-Chase experiment. This is from the bacteriophage standpoint, and which is a T2 phage. We verify the genetic information is DNA. And this layout of the foundation for the getting to the structure of the DNA. So today, at the beginning, we want to mention something about DNA. So what is DNA? Then we'll talk about the detail of the DNA replication. So first of all, what is DNA? It's a very simple question. But some of you may not know the full name. What is the full name of DNA? 2' deoxynucleic acid. That means DNA. DNA, right there. Okay, now what is the basic structure of DNA? Can you draw it? A basic structure is not the complicated, not the fancy one, okay, the basic structure. And we should all know, DNA have a basic components is a pentose. So we call it a 5 carbon. It's called a pentose. And where it comes from? Pentose phosphate pathway using NADPH as a reducing power generate precursor to let the bacteria later on can synthesize DNA. So that's a pentose, 5 carbon. Okay, what are the structure? Is it H? Here is attached to a base. Because a genetic base or gene base, then here you have H, OH, H, OH, H, and this here is CH2. Okay, what is the DNA? So-called 2' dihydroxy. Okay, by the way, this is 1', 2', 3', 4', and 5'. It's right here. This one is gone. Okay, so we write H here. And by the way, this type of configuration, we also call it a beta. Is that right, a beta form? That's most likely is the dominant form in our nature. Then what we're missing here? There is someone here, which is we draw already, is a phosphate. Is that right? So what are the basic structure of a DNA? It's 5 carbon sugar with a base and a phosphates. That's so simple, it's a DNA structure. So what this means, what this tells us? This tells us DNA is a very boring chemical. And you don't think about it so dramatically. When you ever people say molecular microbiology, it's like so fancy stuff. It's a very boring chemical compared to protein. Remember protein, what we have? Amino acids, peptides, primary, secondary, tertiary, is that quaternary structure. It's more prolific and more complicated. And this is a very boring structure. Okay, so now let's talk about that. Then let's talk about something else. Number 3. What is this base? When we talk about here, there is a base. What are these bases for? You should know about these things, is that right? We talk, you learned in your other classes. The base could be looks like a 6 carbon combined with a 5 carbon. So this is a 6 carbon and with a 5 carbon. This is called purine. And they also could be constructed just by a simple 6 carbon. This is pyrimidine. Now, if you know the structure, I don't know whether you like it or not. If you know, you can draw it. It's NH, CH, N, C, C, NH. Then you have NH, C, NH2, NH2, C, NH. And then the carbon starts from 1, 2, 3, 4, 5, 6, 7, 8, 9. And then the pyrimidine, what it looks like is N, CH, NH, CH, CH, and CH. Okay, then you have to have 1, 2, 3, 4, 5, 6. Okay, what are the examples of purine? We have adenine and guanine. What are the examples of pyrimidine? Cytosine and thymine. And you know about these things already. This is called A, this is G, this is C, that is T. Then how they connected with each other? A complementary with T. Is that right? G complementary with C. What it looks like? I'm not going to draw the whole structure, okay? We'll just draw a brief one. Usually I like to draw this way so you can understand more good is in here. Okay, so that's T, that's A, so you have a 2 bonds connected. Okay, this one we can also draw like this. And we have, it's a simple structure, so you understand. This is going to be C, this is going to be G, so, oh sorry, this is going to be C, this is going to be G, so we connected. What are the bonds connected then? Hydrogen bonds. Okay, can we do the review? What are the 3 major chemical bonds what you learned before? H2O, when you have this O, what is this bond? Covalent bonds. Okay, sodium chloride. You have salt, sodium. What is this? Ionic bonds. And right here, you have a hydrogen bonds. Hydrogen bonds is the most weakest bond. That's why they could be hydrolyzed, they could be breakdown. Is that right? Okay, this is lay down the foundation of the DNA structure. We talk about this. Okay, I have to remove this. Okay, so we talk about the DNA already. Now last one. So DNA, we know there is 2 lines. How are they connected with each other? Okay, let's draw here. We're going to use it. Okay, so you have 5 carbon. This is the base. Let's say A, H, 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 O. Then you go CH2, O, P, O. Okay, this is 5 carbon. This is 3 carbon. We go here is P, then go O, and then go CH2. And if we draw another one right here, this is going to be connective here. So we're going to have the T, H, 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 O. And we go here. And we go here, CH2. So we go out here. You can with me draw several, okay? Let's say C, H, 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 O, then we go P, then we go. Okay, so 5 prime, 3 prime. Okay, we go 5 prime, 3 prime. So their connection is always 5 carbon sugar, phosphate, 5 carbon sugar, phosphates, 5 carbon sugar, and phosphates. Always go this order. Okay, now we're going to talk about the big deal. You know that who founded the structure of DNA? In 1953, February, in the University of Cambridge, Francisco Crick, Crick, and James Watson. They published a paper, a 1 page, in Nature. Tells people the DNA is double helix C structure. Right here. You can download from WVU library, 1 page only. It's a serial paper, to be honest. It's not a research-based, it's a serial hypothesis paper by the time. This is people we say it is the single most beautiful article in the 20th century. It's laid down the foundation of the molecular microbiology and biochemistry and genetics. So we want to talk about the story behind it, okay? Francisco Crick, by the time, is a PhD student in Indiana University. And he's doing something related to interested to find the DNA structure, because DNA is already a hot topic by the time. Francisco Crick is still there. If I remember, 28 to, is still there. Okay. James Watson is not there. Okay, very long life. By the time, they have 12 years old difference, but they get along very well. James Watson is a Surgeon General. He's a physician, works for the British Army during the Second World War. And Francisco Crick, when he was PhD at Indiana University, he said, I want to do something. I want to know the DNA structure. So he go to the best university in the world by the time. It's Cambridge University, Cavendish Lab. And at the time, he found James Watson. James Watson's there. One of them, I forgot if James Watson or Francisco Crick, maybe James Watson. His hobby is watching birds. So he had a nickname is Birds Watcher. By the time they do lots of thinking, they do lots of talking, and they do not do much research. And later on the news said Francisco Crick, his people will be dragging the nuts if he's working in the lab. But it's not to saying he's have this big contribution. Now what happened is that by the time when they are talking, when they are thinking about the DNA structure, they get this very important foundation, the double helix is coming from another British scientist, Rosalind Franklin. Very unfortunately, Rosalind Franklin have a very short life. Yes, only 38 years old. Only 30 years old. Rosalind Franklin did a great work, which is published here, but only for acknowledgement. Is a picture right there. What is this called? You learned about this. X-ray crystallographer. And there's a rumor has it, Rosalind Franklin, even she died, she did not know. Her contribution is so big. Why? Francisco Crick and Watson, they awarded the Nobel Prize in 1962. And by the time Rosalind Franklin is already died. Okay, and there is a news said Crick and Watson take her to have a trip in the United States. And when she back to England, she passed away. Very short life, but very talented biochemist. This is the first thing. Second thing, they need another help. A and T, G and C. Is that right? A complementary with A, G complementary with C, with connected with hydrogen bonds. What this means? By the time they meet with another big American chemist, Shawgraf. This comes out with very simple Shawgraf rules. The ratio of A and T and G and C, the ratio is 1:1. So that means in a microorganism or in an organism, it could be a higher amount of A and T, could be higher amount of G and C, but their ratio is always 1:1. And I also want to tell you the A and T and the G and C, they are universal for all the organisms, not only microorganisms, not only human being plants. So there is a joke said, We, all of us, same with bacteria, we are from the same ancestry, an ancestor, because they're all composed by, at the end of the day, we are composed by A, T, G, C. And their ratio and 1 and 1. What this means, what this tells us, this tells us this most likely is correct. Is that right? That's why you have A and T, G and C. Now, Shagraf is a chemist from Columbia University. Later on, he also wins the Nobel Prize later on for other things. By the time he knows A and T, G and C, but he never figured out the structure. And Crick and Watson know the structure, but they did not figure out the gene base, which is the A and T, G and C. So they had a lunch together, and they said, okay, we get it. Shagraf is regret to have the lunch with them, because later on he'll write a story by himself, autograph or something. He said it's a big regret lunch with them, because otherwise he did dig a little bit more, he could find it. Okay? Crick and Watson in February 1953, they published this beautiful paper and find this big breakthrough in the modern history. The first thing what they do, if you have anything happened in England, what they do? They go to the bar. What the bar they call in England is a pub. Is ego pub. So they go there to buy everybody a drink. They say, okay, we find a secret of the life. So everybody have a drink, have a beer. Then he talked about that, but nobody understands. But anyhow, it's come out with this beautiful paper in the world. So that is the story behind it. Okay. Now later on, Francisco Crick think about not only the DNA. So he had the idea the DNA gonna further do something. So he pointed out the original version of the central dogma is a nuclear gas in the two protein. Very similar to this. This is later on what people find it. Okay, so the history is finished. So let's talk about this detailed structure. So we tell you a little bit of history. 1953, Crick and Watson found the double helix of DNA. So we need to talk about that. Double helix of DNA. And I usually need this pen, I break down two. So I'm going to draw a big one. Like this. And this one. Like that. Okay. Not very beautiful, but looks okay. Okay, you learned about that. Minor grove. Major grove. Is that right? One term around 10.5 base pair BP. And the width, this is people say 3.48 nanometer. By the way, when Crick and Watson, they find the double helix of DNA, this is B form. Okay, the most uniform in the natural world, it's not, there's A form, D form, or different type of, this is B form. Now, we know A complemented with T, G complemented with C, A complemented with T, G complemented with C, you can do all these. Okay, what this tells us? What's the implication of DNA double helix? What is the implication? Very simple, you know about it. Semi-conservative replication. What is important for the semi-conservative replication? When you break down, what's going to happen? One line will be like this. Another line will be like this. Okay, once you separate it, sorry, should be go this way. Once you have that, what happened? They will using the mother band as a template, these as a template, to synthesize a daughter band. And the daughter bands ideally should be exactly the same as the mother band, and the genetic information should be transferred exactly, in the exception of some mutation happens, but that happens all the time. So that's a semi-conservative replication. But people have to verify that, because you have this structure. Okay, I'm getting this semi-conservative replication. I need to verify it. It is correct. There comes up another important experiment, Michelson, star experiment. This is around 1955, around. Okay, different book says different stuff. Matt Michelson, Frank Starr did a confirmation study. Semi-conservative replication is working. By the time they are PhD students in a big university, a small private institution, Caltech, it is end up with the most smartest people be there. Did you watch Big Bang Theory? Sheldon Leonard, they are coming from Caltech. The story is there. They are students there. Michelson is still there. Michelson is still alive in the world. Later on, he works in the Harvard University, the professor there. Starr is not there anymore. What they did, and you know about that. Okay, I'm going to add something there. Is that right? I'm going to label them. So, I have a bacterial media. This is the bacterial media. I grow the bacteria. Okay, the mother cell. Originally, that mother cell I'm going to label them with N nitrogen 15. It's not a radiation, just an isotope. Okay, just an isotope. And in the bacterial media, I'm going to pour them M14. It's a normal nitrogen. So when I put there, they have to grow. Is that right? They're going to have to do the replication. What happened? What is the first generation looks like? The first generation, if you wanted to see, the first generation, you will see one line, the mother band is M15, and another one is M14. Okay, the same thing right here. This is M14, the daughter bands, and the mother bands is M15. This is the first generation. If the second generation, what is going to happen? It's going to be more and more M14 and more and more M14, is that right, compared to the M15. So, they started to generate a big research. What they do is in this big tube. This big tube, they are adding a salt, is Sicilian chloride. Once they add Sicilian chloride, they are doing the centrifuge. Once they're doing the centrifuge, what they find? All original mother cell, they are at the bottom. They are N15. And most of the time when they see the daughter cell, they're always on the top. That's a high amount. Later on, people find that maybe after like 5 generations, 75% of them, more than 75% is nitrogen 14, and M15 is only about maybe 10% or less than that. So what you find, this goes here, and this goes here. So this is verify that. Using isotope M40 and M15 and verify that. DNA replication is a semi-conservative replication. Okay, so get down here. Now we're gonna move on to talk about what is semi-conservative replication. Okay, so this is a big story we want to mention in this blackboard right here, and the detail. Also the enzyme, the function of the enzyme, why it looks like that. And what's the story behind it? There's a lot of story behind it, and we could share with you. And it's, oh, I should move this away. The last one. Is useful. Okay, so let's talk about DNA replication. So first of all, DNA replication is semi-conservative. We have two bands. One band's going through 3' to 5'. Another band's going through 5' to 3'. At the beginning, for bacterial cell, everything happened is in this replication fork. They need something to initial. The initial group called DNA group. There is 3 components. We call it DNA A, we call it DNA A, DNA B, and DNA C group. This is DNA protein group. DNA A, basically we say it's an initiator. Initiator, which is started. DNA B is helicase. DNA looks like that. It's a double helix, highly twisted. And it is also negative super coil. Natural. Okay, when they twist a negative super coil. And that super coil you have to break down. So we need to use helicase, which is break down the double helix. Now what is DNA C? This is a helper. And we call it a helicase holder. Okay, these are the 3 things to initiate when they separate. Because DNA is double helix and highly twisted, so from the physics standpoint, they will always keep twisted. So when you separate them, they're gonna very easy to come back. So what we need? We need some protein there, attached to the side. What are these things called? SSB, single strand binding protein. To attach to the separated line to make sure, separate the band, make sure they are separated. Okay, now we start to do replication. Everything's ready, let's say. Now, no matter which band it is, we all need primase to synthesize a small sequencing of RNA primer to initiate the DNA replication. So let's do that. Let's do a give example, okay? I have I usually add a phosphate, P phosphate, G phosphate, C phosphate, A phosphate, T phosphate, G phosphate, and C phosphate. Okay, I add these. This is 3' to 5'. 3' to 5', we know that. They can naturally to do the replication from this way to that way. Because the DNA replication is always from 5 prime to 3 prime. And they need an enzyme. What is the enzyme? DNA polymerase. It's a very good name, polymerization. When we become the chain become long and long, we call it a polymerization. This is the DNA polymerase. DNA polymerase is a group. Have 1, 2, 3, 4, and 5. Here we basically talk about 1 and 3. Okay, and there is a simple description. Some people say they have a different function. But some people say they may just come from a different bacterial cell. So that's why you name them. Now that we know that V4 and 5, this is all used on the SOS recovery. For the mutation recovery. Okay, this is, we'll talk about that later. Here we will talk about DNA polymerase 1 and 3. Now, who is the person find the DNA polymerase? It's American scientist, author Cronenberg. It's a very interesting story behind it, I tell you. Also, Cronenberg recognized it's a DNA polymerase. He found the DNA polymerase. Just recognized. I'll tell you a story behind it. Okay, so the replication is from 5 prime to 3 prime. So we're gonna go ahead to do. So what we gonna to synthesize? Based on complementary policy, this will be from 5 prime to 3 prime. Okay, so what that looks like will be P, PA, PC, PG, phosphate T, phosphate A, and phosphate C, phosphate G. Now, of course, it's going to become long and long, long and long. Okay, it's going to become long and long, long and long because we have these nuclear tides is in the cell membrane system they're going to use to attach to it. Okay? This is key. Okay, now, the first band on the top, we can do natural replication from 5 prime to 3 prime. This is, we call it a leading band. Here comes a question for the second one. By the way, we call it a lagging band. Here the problem comes up. This band is from 5 prime to 3 prime. So we can do it naturally. Now here's the question why. Can you tell me why? There is a true reason why you can't do 5 prime to 3 prime. I should still keep this one here. So let's go back to see right here, the structure. Okay? Let's go back to here, the structure of the DNA. This is what should be CH2, phosphate. Okay, this is 3 prime. Then you go AP. Then you have another one right here. This is connected to CH2. You have A, H, 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 O. Okay, this is O. When you go 5 prime to 3 prime, go this way. There is a free available OH. Okay? So, in other words, go other direction. It's not going to work because there is no free available hydroxy bonds. So you can't do it. You cannot do the other direction. Now, by the way, when every time the new gene base, the new DNA is added, they're going to hydrolyze ATP, and you should know that, okay? So the chain like this. So this is the first thing. From the structure standpoint, there's no free available hydroxy bonds. Hydroxy base. What is the second reason? When you go 5' to 3', you add, and every time a new freely nucleotides with carrying 3 phosphates. When they come in, they usually hydrolyze 2 phosphates. Is that right? This is a very important concept, like you are going to a line. There is a line to go to see WVU with University of Pittsburgh as a football. And you pay the ticket, you join the line. It's very natural. And from the energy standpoint, it's also working. Can we do in the opposite direction? If we do in the opposite direction, what happens? These already synthesized, you have to break down and let you insert. Its energy standpoint is not working, which means the delta G is not going to be negative. Same thing, you're in the line to see the football, you go there, you pay the ticket, like you hydrolyze 2 phosphates, go there. Is that going to happen? Some people, let's say you come in, okay, we're going to pay you. Is that happen often? No. Very few times happen. Is that right? That's same for bacteria. The DNA from the energy standpoint does not work. So delta G is not negative. From the energy standpoint, is not working. Not for 5', not for 3' to 5'. This is a major reason. There's 2 major reasons why this is always from 5' to 3'. Because there is, number one, there is no free available hydroxy bonds in the lagging band, and from the energy standpoint, it's not working. Simply to say, so how are we going to do right here? Well, replication is always from 5' to 3', so we need to synthesize